Blood glucose levels are controlled largely by the endocrine pancreas. Within the pancreas, specialized cells of the islets of Langerhans secrete glucose-regulating hormones into the blood. Alpha cells secrete glucagon, a protein hormone that maintains blood glucose by increasing the release of stored glucose, called glycogen, from the liver. Beta cells secrete insulin, which lowers blood glucose by facilitating the movement of glucose from blood into muscle, liver, and adipose tissue, and amylin, which slows the rate at which digested glucose enters the bloodstream. Delta cells secrete somatostatin. Somatostatin helps regulate blood glucose by decreasing gastrointestinal activity after eating. This delays intestinal absorption of glucose into the blood. To satisfy its energy needs, the body uses glucose, fatty acids, and other substrates as fuel. The brain relies almost entirely on glucose for its fuel. One way for blood glucose levels to rise is by food ingestion. Glucose not immediately used for energy is removed from the blood by the liver, which stores it as glycogen. When blood glucose levels fall, the liver's stored glycogen is broken down and released into the blood as glucose in order to maintain the blood glucose within its normal ranges. Blood glucose levels usually reflect the difference between the amount of glucose released into circulation by the liver and the amount of glucose removed from the blood by body cells. For health to be maintained, blood glucose levels must be kept within very narrow parameters. Cell membranes are impermeable to glucose, so they require a special carrier to move glucose from the blood into the cell. Insulin is that carrier. Insulin is either rapidly bound to peripheral tissues or destroyed by the liver or kidneys. Insulin has a half-life of approximately 15 minutes once it is released into the general circulation. Insulin lowers blood glucose levels in several ways. One way is that insulin facilitates the movement of glucose from the bloodstream into body tissues by binding to a target cell's membrane receptor. The insulin activates the receptors through a protein kinase messenger system, causing an increase in the number of glucose transporter molecules present on the outside of the cell membrane. The glucose transporter molecules, called GLUT4 glucose transporters, facilitate the diffusion of glucose into about 80% of body tissues. Within seconds after insulin binds to a membrane receptor of a susceptible cell, the membrane increases its uptake of glucose. This is particularly true of skeletal muscle and adipose tissues. Another way is that insulin provides for the storage of glucose as glycogen in the liver. When the supply of glucose is abundant, insulin activates the enzyme hexokinase, which in turn phosphorylates glucose. The addition of phosphate to the glucose molecule traps it within the liver cell. Approximately 2/3 of the glucose that is ingested with a meal is removed from the blood and stored in the liver as glycogen. Yet another way is that insulin inhibits gluconeogenesis, which is the building of glucose from new sources. This mechanism of action is a subject of investigation, but appears to involve the promotion of hepatic glycogen cycling, as well as the interaction of insulin with transcription factors. And finally, insulin also reduces blood glucose because it facilitates triglyceride synthesis from glucose in fat cells, while inhibiting the intracellular breakdown of stored triglycerides. The role of insulin in blood glucose control functions by means of a negative feedback cycle. The cycle begins with the body's blood glucose level increasing. In response to a higher level of blood glucose, the pancreas's beta cells release insulin into the blood. The insulin binds to insulin receptors present on most body cells. Once bound, the protein kinase messenger system causes an increase in the number of glucose transporter molecules, which then facilitate diffusion of glucose into the cell. When glucose is carried into the cell, it decreases the level of glucose in the blood, and this negative feedback inhibits further stimulation of insulin release. Alpha cells then respond to a lowering of blood glucose by increasing the secretion of glucagon, which stimulates gluconeogenesis, the process by which the liver turns its stored glycogen into blood glucose, maintaining blood glucose levels between meals and during periods of fasting. Glucagon, because it counteracts the functions of insulin, is sometimes called a counterregulatory hormone. Other counterregulatory hormones include the catecholamines, which help to maintain blood glucose levels during periods of stress by inhibiting insulin release from the beta cells. Growth hormone, which decreases cellular uptake and use of glucose, resulting in an increase in blood glucose levels. And the glucocorticoid hormones, which stimulate gluconeogenesis by the liver. Fasting blood glucose levels are normally kept within a very narrow range. When blood glucose increases above its normal level, insulin secretion from the pancreas increases rapidly, then returns to baseline in 2 to 3 hours. A plasma glucose level higher than the normal fasting blood glucose level is defined as hyperglycemia. Hyperglycemia is usually caused by insulin deficiency or results from decreased cellular responsiveness to insulin. An abnormally low blood glucose level is defined as hypoglycemia. Hypoglycemia can be caused by fasting, or especially by fasting coupled with exercise, because exercise increases the usage of glucose. However, it is more likely to be due to taking glucose-lowering medications without enough food. Glucose is the energy substrate used most immediately by the body, especially the nervous system. The endocrine system performs the vital function of maintaining blood glucose levels within normal, narrow parameters. The beta cells of the pancreas produce the most important glucose-regulating hormone, insulin. An imbalance of insulin and other hormones can lead to abnormally high blood glucose levels, hyperglycemia, or abnormally low levels, hypoglycemia.